Next speaker is one of the two contributed talks by Sofia Freitas, PhD candidate in Alfonso Renard's lab here at Champalimaud, and Joe Patton. And she's going to be talking about temporal control at multiple scales.
Okay, thank you for the introduction. So the environments in which we and other animals have evolved contain rich temporal structure. And regularities in that structure shape the temporal control of behavior, and indeed behavior at multiple scales. And today I'm going to tell you about my recent work focusing on behavior at the relatively short time scales of seconds to minutes. Even at these time scales, there are potentially multiple distinct forms of temporal control. For example, a surfer attempting to catch one of these waves must estimate its arrival time to determine when to start paddling, and then modulate the frequency and intensity of each stroke in order to be in a position to descend the wave's face as it breaks. Now, in the laboratory, we can devise behavioral tasks to bring such processes under experimental control and study their features quantitatively. The task I'll be describing today is based on a classic paradigm and involves operantly conditioning rats to press a lever for fluid reward under the so-called fixed interval schedules of reinforcement. The task goes as follows. At the onset of the fixed interval, a clock is started that counts down until the fixed interval has elapsed, at which point the rat can press a lever to produce a reward. Now, though pressing the lever before the fixed interval has elapsed produces no rewards, animals typically transition to a state of repeated pressing, even before the interval has elapsed, and maintain that state throughout. Until a reward is produced, at which point a new clock starts and the process begins anew. So reward is available periodically, contingent on rats pressing the lever. And this task itself allows us to extract two straightforward measures of temporal control at distinct scales. The time of transition to a state of sustained pressing behavior, which we'll be referring as the transition point, and the frequency with which the rat presses the lever within that state. We will then systematically vary the duration of the fixed interval across blocks of trials, as well as other task features, which I will describe in a moment. We also, of course, track other behavior in a more continuous manner using high-speed video, but I will not be presenting that data today. Previous work from our group and others suggests that these processes, that the processes underlying these two forms of temporal control may be at least partially dissociable, as certain manipulations of the task may affect one but not the other. And these are results that we will build on in the next few slides. So the first thing I want to point out about the rat's behavior is that the distribution of these transition points, so between the state of non-pressing to one of sustained pressing, scale proportionally with the duration of the interval. Here I'm showing probability density functions of transition points measured during fixed intervals of 15, 30, and 60 seconds. If you can see that the longer the fixed interval, the more delayed and broad the transition distributions become. However, if I normalize the time axis to the fixed interval, these distributions become completely overlapping, demonstrating scale invariance of transition times relative to the fixed interval. The data I'm showing here was acquired under conditions that the reward magnitude was held constant while the fixed interval was allowed to vary in blocks. Which results naturally in a changing of reward rate systematically with the fixed interval. And one might have expected that under the higher rates of reward associated with shorter fixed intervals, rats would be more motivated to start pressing the lever sooner, but our data indicates that that's not the case. But instead, rats transition to pressing at a similar time point relative to the fixed interval, but assumed pressing rates that varied with the fixed interval. Now, to test whether differences in pressing rates were due to the impact of changing the fixed interval itself or to differences in reward rates, we can vary the magnitude under a given fixed interval, in this case 30 seconds, which indeed leads to higher rates of pressing for larger rewards, but once again without affecting the timing of transitions. Lastly, we can vary the fixed intervals, as here displayed on the left, but now we vary also the reward amount, giving larger rewards for longer intervals to approximately match the rate of rewards across interval durations. which leads to approximate equalization of the pressing rates without once again affecting the timing of transitions. So this behavioral data represents a double dissociation between the effects of these two distinct task features, the fixed interval duration and the rate of reward, and these two different forms of temporal control of behavior, i.e. the timing of transitions into a state of pressing, and once in that state, the intervals between successive presses or pressing rate, which we take as our measure of action vigor. Now, in particular, I just showed you that we found that the timing of action initiation was affected by the duration of the interval, but not by reward rate. And conversely, the vigor of action execution was strongly modulated by reward rate, but not affected by interval duration per se, as long as reward rate was kept approximately constant. Okay, so with these experimental levers and these two forms of temporal control, we're now going to look into the brain to try to understand how these might be produced. One compelling place to look, and where we will be looking today, is the basal ganglia, because a large body of work over many years has led to the view that it plays a critical role in learning programs or policies for action selection. Its main input structure, the striatum, receives rich input from cortex and thalamus that provide information about the situation or state that the animal finds itself in, as well as information about the actions on offer in that situation. Now, that state and action information converges with extremely dense dopamine input from midbrain, and dopamine itself is a neuromodulator thought to convey a teaching signal from computational models of reinforcement learning called reward prediction error. Another interesting thing about this architecture is that there are two main feed-forward circuits through the main axis of the basal ganglia, initiated by so-called direct and indirect pathway striatal neurons, that, because of their downstream circuitry, decrease or increase inhibition on targets of basal ganglia output. And because these two pathways express different dopamine receptors, the effect of this dopamine-driven learning system appears to be one, to promote actions that yield better than expected outcomes, while suppressing those that yield worse than expected ones. Indeed, a very elegant system that evolution seems to have arrived at. Now, to perform its presumed function of learning effective policies, this system will require access to temporal information in order to appropriately implement these policies in time. And indeed, a large body of work from many groups, including ours, has revealed a critical role for both dopamine and striatal population activity in the temporal control of behavior. Specifically, it is relatively well established from clinical to laboratory settings that higher dopamine levels correlate with higher movement vigor, whereas its role in timing is a bit more varied in the literature. Striatal dynamics, on the other hand, seem to evolve along some trajectory state with a speed that correlates with temporal judgments, whereas vigor seems to be represented along some other neural dimension, namely at the level of a gain signal, presumably being implemented by direct pathway activity. Now, given these roles in different scales of temporal control and this anatomical background we just went through, there are hints that these two systems might influence each other and even operate in parallel. But as far as we can tell, dopamine and striatal activity have historically been measured separately. And you can probably see where I'm going with this. So to address this issue, we simultaneously recorded dopaminergic activity using fiber photometry and electrophysiology activity using neuropixels in the striatum of rats performing our task. Now, what I'll show you next are some very preliminary, but we believe nonetheless exciting results. And some of what I will show then might also change as the data set grows, so keep that in mind. In the interest of time, I won't go over all the results, but I'd like to briefly highlight a few pieces of evidence suggesting that these systems are indeed involved in temporal control at the scales that we're interested in. So for instance, when we varied the fixed interval duration, dopamine signals evolved over time in a ramp-like fashion that appeared to be specific to the interval being timed. Turning to the striatum, we can recapitulate findings from our group showing that striatal population activity unfolds in a non-repeating pattern throughout the interval, consistent with a clock-like signal. And I hope you can appreciate that across these two examples of fixed interval durations of 30 and 60 seconds, the population dynamics scale appropriately to each time scale, while maintaining the same relative ordering of the neurons. The next result I'd like to share takes advantage of something unique about this data set, which is the fact that we have simultaneous dopamine and electrophysiology recordings. Now, because neurons in the direct and indirect pathways express distinct dopamine receptors that are differentially modulated by dopamine, in which direct pathway neurons are driven by high dopamine concentrations, whereas indirect pathway neurons by low dopamine concentrations, this potentially opens up the possibility of classifying neurons into putative pathway populations based on how their activity co-varies with dopamine. I should note that, again, this is preliminary, and it's still exploratory and subject to change, but it's a promising avenue, and what I'm presenting it here primarily as a proof of concept of what might be possible with these kinds of combined data sets. And so, to do this, we computed spike-triggered dopamine averages for each neuron. The way we do this is, for every spike, we take a short window of the dopamine trace around it, and then we average all the snippets of all the spikes for that neuron, essentially getting a spike-triggered average. Here, an example of a cell with its spike raster aligned to the transition point. Allow me to show a few more examples. And to a first approximation, we basically observe that there are two broad groups of cells. Those with whose spikes tended to coincide with increases in dopamine, and those associated with decreases, namely in blue and in red. And to capture this a bit more systematically, we extracted several features from the spike-triggered dopamine waveforms and applied a nonlinear dimensionality reduction that we then clustered to identify potential functional subpopulations, which were identified as being putative indirect pathway cells, labeled in red, and putative direct pathway cells, labeled in blue. The examples that I'm showing here were matched for firing rate and recorded within the same session. And what's interesting is that even from these initial examples, we can see a clear difference in the activity patterns around the transition point. Putative indirect neurons tend to be active before the transition and then fall silent, whereas direct pathway neurons show the opposite pattern, low activity before the transition and the rise afterwards. A pattern that almost seems to point out that to bring about the transition, there's a clutch-like mechanism in place in which the action that is happening needs to be suppressed and then followed by promotion of the subsequent action. Now, for this next part, we focused our analysis on the 30 seconds fixed interval and took advantage of the natural variability in behavior to probe these two scales of temporal control. To do this, we sorted trials into terciles based on the transition timing from early to late and pressing rate from low to high, and examined how neural activity and the putative, direct, and indirect pathways varied across these behavioral categories. When conditioning on transition timing, we observed that putative indirect pathway neurons dominated the early part of the interval, showing high activity before gradually decreasing, and while putative direct pathway neurons showed the opposite pattern, with activity increasing as the transition approached. And importantly, the crossing point between these two pathways occurred close in time to the behavioral transition itself, and this alignment was consistent across the different timing terciles. When conditioning on vigor, we found a distinct modulation pattern. Before the transition point, lower vigor was linked to greater pathway separation, mainly due to stronger inhibition from the putative indirect pathway, whereas higher vigor corresponded to a smaller difference between the pathways. To wrap up, I've shown you that even with a relatively simple operant task, we can distinguish two complementary forms of temporal control: the timing of action initiation and the vigor of the subsequent action execution. Behaviorally, we found that the timing of transitions into sustained pressing scaled systematically with the duration of the fixed interval, whereas the vigor of pressing scaled with the rate of reward. Turning to the neural data, I showed you some preliminary analysis pointing to a neural separation that mirrors the behavioral one, with the timing of behavior transitions being correlated with differences in the putative pathways balance and vigor being reflected in systematic changes in the relative difference between these two pathways. Of course, these are early observations, but they provide an encouraging proof of concept that this combined dopamine electrophysiology approach can begin to reveal how basal ganglia circuits contribute to the temporal organization of behavior. With this, I'd finally like to thank both my labs, especially my supervisors who were very patient with this talk and throughout the PhD, and my collaborator, Mafalda. That's it.